Hello, everybody. Today we shall be discussing the various vectors that are used in giving effect to the recombinant DNA technology. That is, those vectors which carry the desired stretch of DNA to the cloning site. They form multiple copy of the desired DNA segment or DNA segment of our choice or gene of our choice, and then those multiple copies may be used for the expressive purpose or for the cloning purpose. That is, they may be used for the expression of those genes or for making multiple copies of those genes, such as cDNA library or genomic library and all that. So the vectors are very important, and there are numerous vectors, and we choose vectors depending upon our needs. So what is the stretch of the DNA? How longer is the stretch of DNA that is to be carried? And what has to be done with that stretch of DNA? So the first we should see the vectors, that how many types of vectors are there. And then while dealing with those vectors, we will see that how genes or the DNA stretch which has to be cloned is integrated into those vectors, and how those genes are multiplied, and how the transformed bacterium, that is the bacterium or the other host which has taken our desired stretch of DNA, could be identified from other such bacteria. So these are the problems which we will have to face while developing and propagating a vector. So let us begin with the slides here. So today we are dealing with another section of the tools and techniques of recombinant DNA technology, and that section will be dealing with the vectors which are an important tool by which we propagate that stretch of DNA to the different organisms which harbor those desired stretch of DNA along with the vectors. So let us begin with this selection. This section will be dealing with following aspects of cloning vectors. So what is the definition? What are the vectors? How will you define the vectors? And what are the features of the vectors? Then what are the types? Say, for example, plasmids, plasmids, bacteriophages. Bacteria are artificial chromosome, artificial chromosome, and human artificial chromosome, to enumerate a few. So in this section, we will deal with the plasmids and bacteriophages and chimeric vectors developed through the combination of plasmids and bacteriophages, such as phosmids, etc. And in the next section, we will deal with the artificial chromosomes, that is AC, BAC, YAC, or HAC. So here is a slide of academic interest, that vector system, host cell, and insertion capacity. That is, utilizing this vector, that is the utilizing plasmid as a vector, how long segment of a DNA can be propagated, how long stretch of a DNA can be propagated. So these data are in kilo basis. So Using plasmids, you can propagate a DNA stretch of 0.1 to 10 kilobases, bacteriophages 10 to 20, so on and so forth, phosmids, bacteriophage P1 and artificial chromosomes, etc. So let us begin with the definition of vectors. How will you define a vector as far as the recombinant DNA technology is concerned? So vector is a fragment of DNA. It is a stretch of DNA, small stretch of DNA, which has the capacity to insert a longer stretch of DNA, which is to be propagated by the recombinant DNA technology. So vector is a propagation vehicle of the DNA, which we are interested, that is, which we are interested to propagate, that is, of which we are multiple copies, trying to form multiple copies. So the stretch of DNA in which we are interested, which is of our choice, should be propagated by these vectors. So these vectors act as vehicles. So an important characteristic of vehicle is that it should be not clumsy, it should not be very long, rather it should have accommodative space so that it could accommodate a very longer stretch of DNA or a number of different fragments of DNA, so that in one go we can clone so many stretches of DNA which we are desired to clone. So the vector may be classified as cloning vector and expression vector. In cloning vector, we make multiple clones, multiple identical copies of that DNA fragment. Clone is genetically identical copy. So if we have a stretch of DNA and we are trying to obtain so many copies of that DNA, then we will go for the cloning vector. And if we are trying to have the product of that DNA, then we will go for the expression vector, which will lead to the expression of that DNA, and the end product encoded by that DNA could be harvested by the expression vector. So what are the properties of good vectors? Good vectors should be, now there are several regions in the vector. Say, for example, a vector should be able to make its own copy. Okay, if it could not make its own copy, then we will not be able to obtain so many copies of that DNA which we have inserted into the vector. So for making its own copy, it should be a ori site, that is the site of origin of replication, so that it could replicate into a number of similar entities. Then there is a selectable marker. It should have selectable marker. So what is selectable marker? Selectable marker is a marker by which we are able to identify the vector which is flourishing in a cell having recombinant DNA technology. That is, suppose we have propagated X segment of DNA into E. coli. So E. coli will multiply and it will make many copies. So how could we identify the E. coli cell, that particular E. coli cell, which is having that stretch of our desired DNA? And to identify, there should be a marker, and that marker is known as selectable marker. It could be antibiotic resistance, and so many other markers are also there that we will see in the subsequent slides. It should have unique restriction enzymes. We have seen earlier that the restriction enzymes are the seizure of genes or the cutter of desired stretch of DNA. So we vector should have sites for action by the restriction enzymes. Say, for example, we have restriction enzyme EcoR1, and if we cut a fragment of EcoR1, which is to be propagated through a vector, there should be a site for EcoR1 in the vector too. And so when we are able to cut these two entities by one seizure, only then they will have sealable ends, that is, they will have sticky ends, and those sticky ends will coalesce with each other, readily coalesce with each other, and then we will apply the glue, that is the ligase enzyme, which will impart permanence to the union. So it should be site of origin, it should have selectable marker, it should have unique restriction enzyme site, relaxed control so that multiple copies could be formed easily. Then there is a shuttle vector. This vector can be expressed into different mediums. Say, for example, there is a shuttle vector which could be expressed in E. coli as well as yeast. So this vector has origin site, ori site of E. coli. If it is a plasmid, then it has got the ori site of the plasmid, as well as it have automatically replicating sequence ARS site of yeast2, so that it could multiply in the bacterium as well as in the yeast. It should have centromere of the yeast so that it could divide into the yeast. Similarly, it should have selectable marker of the bacterium. Say, for example, if it is PBR 322, then it should have ampicillin resistance as well as tetracycline resistance. Similarly, in bacterium, it should have uracil marker so that it could be in yeast, it should have uracil marker so that it could be identified easily there too. So these are the characteristics we incorporate in shuttle vectors. We incorporate the features of both the vehicles, both the flourishing sites, both the flourishing cells. Say, for example, one cell is E. coli, another is yeast. So we should have that vector should have important site so that it could divide into the E. coli, so origin site, so that it could be identified in E. coli, so the selectable marker. Similarly, if it is flourishing yeast, it should have ARS sequence, automatically replicating sequence, as well as it should have centromere so that it could divide, it could replicate, as well as selectable marker too, so that the yeast transformed yeast, that is the yeast having the desired stretch of DNA, could easily identify. So this is about shuttle vector. Now there are polylinker. Polylinker is the multiple sites for integration of different types of desired DNAs. That is, when we are preparing recombinant DNA, we integrate DNA of our choice into a vehicle, into a vector. So polylinkers have poly sites for integration, and so they are also called multiple cloning sites. They should have multiple cloning sites, also called polylinkers. It would be better to call multiple cloning site because it is self-explanatory. It has got so many sites for multiplying the desired stretch of DNA, or for introducing or integrating the desired stretch of DNA into the vector. So the vector having multiple cloning sites or polylinkers are very useful, and one such vector which had been artificially designed is PUC18. So we shall see what are the important characteristics of PUC18 in our later slides. So here we have got some important characteristics of the vectors. They should have site of origin so that they could multiply. They should have selectable markers so that the transformed cells, that is the organisms which have got the characteristics of the integrated foreign DNA, could be easily identified from the other normal organisms. So they should have selectable markers, they should have relaxed control over replication, and they should have multiple cloning sites so that many stretches of DNA could be integrated in one vector itself. Now, coming to the plasmids. Plasmids have been employed as vectors in a plethora of cases. That is, the plasmids are most common vectors. So we should see what plasmids are. You might have heard plasmid a number of times. I don't know whether you know its characteristics or not. Let us have a quick recap that what are the plasmids. Bacterium contain its chromosomal DNA. That is the circular DNA in most of the cases, which is the main, which represent the main chromosome of the bacterium and which replicates as the bacterium multiplies. But bacteria also contain, many of the bacteria have extra chromosomal DNA, a stretch of extra chromosomal DNA, which can multiply autonomously of the main chromosome. That is, they are not the part of the main chromosome, they are residing beside the main chromosome, and they have the ability to multiply too. That is, they replicate and form their own copies. So they have site of ori, that is site of origin of replication, and they can multiply. There are some such extra chromosomal DNAs which can get integrated into bacterial chromosome too, and these are episomes such as the F factor or the sex factor of the bacterium, which can get integrated into the bacterial chromosome, as well as which can exist as an independent autonomously replicating entity. So they have tra-site for transfer, F plasmid in case of F plasmid. They have rep regions for controlling replication. Insertion elements impart episomal function. That is, there are insertion elements. So insertion elements are made up of a few nucleotides, and they border a particular stretch of DNA, and they empower that stretch to function in a particular way. So insertion elements, you can say, are akin to transposons in many ways. So insertion elements impart episomal function. Tra imparts ability to promote its transfer. So these F factors are transferable in case of conjugation. So if F positive bacterium conjugates with F negative bacterium, a copy of F factor will be transferred from the F positive to F negative, and the recipient, that is both the X conjugates, including the donor and the recipient, will be F positive. That is, each of them will have one copy of F episome, and the F episome have genetic markers which code for the synthesis of F pili, that is the finger-like projections on the surface of the bacterium which make contact with another bacterium, and conjugation tube is hollowed out of them, and transfer of F factor takes place through the conjugation bridge. Then there are R plasmids, which impart resistance to the host to the antibiotic and growth factors, possess drug-resistant genes, and here came the transposable elements, and spread through natural population. Transposomes are jumping genes, which we would be able to discuss in some another class. So virulent plasmids, these plasmids cause pathogenicity to the host cell. The host cell will be adversely affected by those plasmids. Then TI plasmids, very important plasmid of Agrobacterium tumefaciens. These TI plasmids have been termed as the natural case of genetic engineering because it transfers genes from one to the other. So Agrobacterium tumefaciens have got TI plasmids which causes gall formation in angiospermic plants. So this is the plant plasmid. Then cold plasmid confer ability to host to kill other bacteria. So these plasmids provide selective advantage to the bacterium in which they reside. That is, they synthesize such type of substances which inhibit the growth of its competitive bacterium or altogether kills the competitive bacterium, so that the plasmid which is resident in a particular host will have advantage for that host which is harboring that plasmid. So there are degradative plasmids too. These plasmids degrade have degradative capacity of complex chemicals such as toluene, and we have made giant bugs by combining different plasmids through which petroleum, which is not naturally easily degradable, can also be degraded. So plasmids could be used, and we are trying to use these plasmids by their genetic engineering by synthesizing a plasmid which is able to degrade non-biodegradable non-biodegradable chemicals. As so, in case of treating pollution also, these plasmids have got tremendous importance. Now, let us deal with the selfishness of plasmid. So this is very important because once plasmid sets its foot in a particular cell system, it inhibits the entry of another plasmid. That is, the plasmid, when it gets the stranglehold, or you can say when it can get a stronghold inside a bacterium cell, it will try to inhibit the entry of another plasmid, and so they tend to exclude others from flourishing in the same bacterium. Two plasmids of same compatibility group cannot flourish in the system, and these are called incompatible plasmids too, and they are having same in same bracket as in same compatible group too. So let us decipher this paradox, that how are they are compatible as well as incompatible. So here we have two instances: cell cycle with plasmid, one plasmid, cell cycle with two plasmid. So here there is only one plasmid. This plasmid replicates itself, and two copies are formed while the cell is dividing, and one plasmid each is distributed to each of the two daughter cells. But here in cell cycle 2, here are two plasmids. When the cell grows and it is dividing, none of these two have made its own copies. Okay, so they should have replicated to make their own copies, so that one copy should be transferred to one daughter cell and one to the another daughter cell. So neither of the two have replicated themselves, and that's why one is distributed to one daughter cell, whereas the another is transferred to another daughter cell. So why this lack of replication? It has been seen that these two plasmids employ the same strategy of replication, that is, their oocytes are having the similar, you can say, makeup, so that the cell could not easily distinguish between these two, and the cell is not triggering their replication. So the cell is confused because of having the same strategy of replication of the two plasmids. So in that sense, they are compatible because they adopt the same strategy of replication, and the oocytes are similar, so that the cell gets confused and their replication is not triggered off. That's why the two remain unreplicated, and they are distributed in one of the two daughter cells. That is, they are not kept together. So they have become incompatible. They could not exist together because of their replication strategy, because of their replication site, because of their oocyte, which confused the cells. That is, the cell could not take a decisive action to make their copies, and since they have not made their copies, they are transferred in an isolated way. That is, one is transferred to one cell of the daughter cell, and the second is transferred to another. So this is how the incompatibility of the plasmid functions, and that's why sometimes it is said that the plasmids are selfish entities. That is, they do not allow the another plasmid to harbor while they remain inside a particular cell. Now, the most common vector plasmids have been utilized, as I have said earlier. Plasmids have been utilized as vectors in a big way in genetic engineering, and which is the common name for recombinant DNA technology. So, in fact, at the beginning of the recombinant technology, we used plasmids, and we learned the steps of recombinant DNA technology through the plasmids. So the most common plasmid which have been used in case of recombinant DNA technology is PBR. So P, the naming of plasmid, you can see, P stands for plasmid, B for Bolivar, R for Rodriguez, and 322 is the number. So P is plasmid. This BR represents the lab in which that plasmid has been developed, and this is the number, the serial number of their identification or development. So this is plasmid. It was developed in the lab of Bolivar and Rodriguez, and so this BR, and now this 322 represents the number. So the plasmid vector PBR322 found in E. coli is the most common one. The P stands for plasmid. BR tells us in the laboratory in which they have been developed. So this part of the vector name stands for, as I said earlier, Bolivar and Rodriguez, the two scientists who constructed the PBR cloning vector, and number 322 indicates the serial number of discovery. So we are most accustomed with these plasmids that is the PBR320. So it is double-stranded DNA closing plasmid. It has got oocyte, it has got selectable markers too, and it has got site of integration of desired DNA. So you can say it is an ideal and simple plasmid through which we can propagate a particular stretch of DNA. So This could confer antibiotic resistant tetracycline and ampicillin, resistance. And there is one plasmid developed from PBR 322, is PBR 327, in which a certain segments, say of 1089 base pairs of nucleotides have been removed between 1427 and 1525-16. So 1089 nucleotides have been removed from the PBR 322 to create a bigger slot for integration of a longer stretch of DNA, and this plasmid is called 327. It has been a modified version of 322 plasmid. So in the next slide, here you see an Ardell plasmid vector. So you see here is a selectable marker. So it may be antibiotic resistance, it may be antibiotic resistance, it may be some other selectable marker that we shall see later on. Here is genes for antibiotic resistance here too. Here is the promoter site, which promotes the replication. And here is the site for restriction endonuclease. So these sites are cut by the restriction endonuclease of a particular type, and the DNA fragment cut by that particular type of restriction endonuclease could be easily integrated into this region. And here is the origin site. Here, origin of replication takes place. So these are the basic features that a plasmid should have. Now, we can see that how these basic features are being reflected in our PBR 322. So here you have site of origin. Here you have one marker, ampicillin resistance. Here you have got another marker, tetracycline resistance. So ampicillin, tetracycline are two markers. Here in this case, you can see that these are MH1 in 3. These are the integrative sites. That is, these are the sites which could be cut by these restriction endonucleases, and a foreign DNA and aligned DNA, which we are trying to propagate, could be integrated in this segment. So let us see it in detail, that how it acts. Say here you have got tetracycline resistance site. Now there are sites of integration, sites of action, specific sites of identification by a particular restriction endonuclease. So if we cut this site by restriction endonuclease and we integrate another DNA, so here we have cut that site, and now we have integrated the DNA of our choice here. So due to the integration of this DNA, what will happen? Due to this integration of DNA, this site, which was providing tetracycline resistance, will be disrupted. Okay? So due to the disruption of this cycle, this transformed plasmid will not be able to impart tetracycline. It could be identified only by ampicillin resistance. So ampicillin is very much here. So you have got ampicillin resistance, it is intact. So here this selectable marker will be ampicillin resistance as well as destabilization of tetracycline resistance, because we have incorporated selectable the stretch of DNA which was to be selected, which was to be multiplied, which was to be cloned in this site, and that's why the disruption has taken place of this site. That is, it represents the tetracycline resistance. So tetracycline resistance will be lost, whereas the ampicillin resistance will be performed, whereas in unaltered, that is those plasmids which are not having the DNA of our choice, they will exhibit both tetracycline as well as ampicillin resistance, and that's how the selectable marker will indicate that this organism has got selectable marker will indicate that this organism has got the DNA of our choice, and the organism which is showing resistance to both ampicillin as well as tetracycline shows that they do not have the stretch of DNA which we have integrated. That is how we could select between the altered, that is the transformed plasmid, as well as the original plasmid. Now we were talking about the plasmid having polylinkers, that is multiple site of integrations. So we have devised another plasmid, which is called PUC18. So it contains antibiotic resistance gene to screen the host. It has got antibiotic resistance as well as some other resistance too. So here you see that it has got ampicillin resistance. Okay, this is ampicillin resistance. And here you have got a LAC-Z gene also. This LAC-Z gene produces an enzyme called beta galactosidase. Beta galactosidase. So we have a marker which produces beta galactosidase. This beta galactosidase will act upon a substrate. This substrate is BCI G, that is bromochloroindoxyl galactoside. It is bromochloroindoxyl galactoside. So this substrate, which is commonly called XGAL, so its name is XGAL. So we have XGAL, which is bromochloroindoxyl galactoside, bromochloroindoxyl galactoside. Now, acting upon this beta bromochloroindoxyl galactoside, the beta galactoside transform it into bromochloroindoxyl bromochloroindoxyl. So beta bromochloroindoxyl galactoside is transformed into bromochloroindoxyl by beta galactosidase, and this chemical goes dimerization. This chemical goes dimerization. It undergoes dimerization, and it will form indigo. And this indigo will give the solution dark blue. Okay, so we have bromochloroindoxyl glycol. We have this bromochloroindoxyl galactoside, and this is being broken down by beta galactosidase into bromochloroindoxyl, and this is 5-bromo-4-chloroindoxyl, and dimerization will lead to dibromo dichloroindoxyl, and that is indigo will be produced, and this indigo will give rise to the blue color. So why was I discussing all these in so detail? Because you have got this site. Let us have the pen. So you have got this gene, lac-Z gene. If lac-Z gene is present, then the animal, the organism, that is the E. coli, which is harboring this PUC18 virus, in presence of X-gal, will give blue color. That's all. Because BCID will be transformed into BCI, and BCI, dimerization of BCI will give rise to the indigo, and indigo will give blue color. So in this case, where there is no so here you have not disrupted the lac-Z gene. But now you see that lac-Z gene also has got many restriction sites too. Okay, so these restriction sites will lead to the incorporation of GNA of our choice. So alien DNA will be integrated. Where it will be integrated? In the lac-Z gene. Now we can see here that this alien DNA has been integrated where? At the lac-Z site, because it has got so many restriction sites. So when it gets integrated into the lac-Z site, it will lead to the disruption of the lac-Z region, and destruction of the lac-Z region will lead to the restrict. Absence of beta galactosidase. So beta galactosidase will be produced by lac-Z gene, and since lac-Z gene had been disrupted by integration of the DNA of our choice, will beta galactose be there? No. Since it has been disrupted, beta galactose will be also disrupted. That is, the production of beta galactose will be disrupted altogether. Now, when beta galactose is absent, will this BCID or X-gal will be converted into bromochloroindoxyl? No. When bromochloroindoxyl will not be produced, there is no question of its dimerization. So there is no question of its precipitation and blue color formation. So mind my words. When we have got unaltered lac-Z site, that is, in case of normal plasmid, when we have not integrated the DNA of our choice, what is happening? That lac-Z gene is producing beta galactosidase, and this beta galactosidase is acting upon a substrate which is present there. It's bromochloroindoxyl galactoside, and it is transforming this bromochloroindoxyl galactoside in bromochloroindoxyl, and two molecules of this BCI will form indigo. That is, and indigo will give rise to the blue color. So in normal circumstances, when we have not integrated the desired stretch of DNA, the lac-Z gene is intact and blue color is being seen. But when we disrupt the lac-Z gene by integration of DNA of our choice, beta galactose will not be produced, and so blue color will not be obtained. So how this marker is being used? Presence of blue color, that is, there is no foreign DNA. Absence of any color, that is, the plasmid is having an integrated stretch of DNA, that is, it is a recombinant plasmid. It has got transformed. It is not synthesizing the beta galactose anymore. And that's why this beta galactose, this beta galactose is a selectable marker. It is through this beta galactose that we are able to select the transformed plasmid or the organism if beta galactose is intact. That is, if no foreign stretch of DNA had been integrated, it will synthesize beta galactose, and beta galactose will transform BCIG ultimately into the indigo, and that will give us dark blue color. But if the beta galactose had been disrupted by integration of the foreign stretch of DNA, then the beta galactosidase will be absent, and so BCIG will not be transformed anymore, and no color will be obtained. So that is how this selectable marker is being used in PUC18, and that selectable marker, ampicillin, is also there. But this lac-Z site, which codes for beta galactosidase, is very important. Now, let us see that here you have got the polylinker. There are so many sites, contains many restriction enzyme recognition sequences. Here you have got many sequences, and the tetracycline resistance is no more there. And the polylinker is inserted with its lac-Z gene. So here you have got the polylinker, that is multiple cloning sites. And in this multiple cloning site, we have incorporated the LAC-Z gene. And so this LAC-Z gene is imparting selectable characteristic to the transformed plasmid. So XGAL supplied in the medium, XGAL is BCID, bromochloroindoxyl galactoside. So this, when it is supplied in the medium, bacteria with wild type LAC-Z produces blue colonies because LAC-Z secretes beta galactosidase, and beta galactosidase will transform XGAL in the indigo, and that will give blue colonies. But when you have got polylinker sites here, so when we integrate the gene of our choice, LAC-Z function will be hampered, and so no blue color will be obtained. And this vector is useful because it have as much as 18 restriction endonuclease site. So it could be acted upon by 18 restriction endonucleases, and that's why 18 different types of fragments could be integrated into this polylinker region, and it will act as a transformed or a recombinant DNA in a bigger way than the other plasmids. So new vectors based on M13 and PUC vectors with new restriction site continue to be developed. This is an ongoing process. So we have developed these plasmids. We are on way to developing another more advanced plasmids by taking lesson from our earlier experiences. Now this bacteriophage, we were dealing with the plasmid and vector as vectors, and we are now dealing with bacteriophages and the vector. And here you have got one lambda bacteriophage. So there is one characteristic about the lambda bacteriophage, that it undertakes both types of life cycle, that is lytic life cycle as well as lysogenic life cycle, which is also called temperate infection. So in the lytic life cycle, what happens that when the lambda phage enters into its source, it multiplies itself and causes cell lysis, and the new phages are released which affect new hosts, which infect new hosts, and so the multiplication goes on. But in the presence of a gene product which is called CI, which inhibits the normal lytic cycle, what happens that this lambda phage gets integrated into the host genome, that is the genome of the bacterium. It is a bacteriophage, so obviously it thrives upon the bacterium. So when it enters the lysogenic phase of its life cycle, that is, it causes temperate infection. It integrates itself, its genome, into the host genome and multiplies as if it were a part of the bacterial host genome. So for that particular part of time in which it is undergoing lysogenic life cycle, its multiplication takes place along with its replication takes place along with the host DNA, and no lysis takes place. And that integrated phage is called prophage. So the bacteriophage can lead its life cycle as an independently multiplying bacteriophage DNA, which becomes covered by the protein, matures, causes cell lysis, and then infect another healthy host and causes cell lysis of that host. And in course of lysis of cells and cells, it could multiply itself into a number of times and build up its population. Another case is that it enters into the host genome, lies quiescent there, and it multiplies itself as the multiplication of the host genome takes place, and that's how it makes its copies it gets transferred into the daughter host cells, but it does not causes the lysis. So that was one important part of the life cycle, which we will have to take into account while we are devising lambda file as the vector. Another important characteristic of lambda file is that 12 bases single-stranded sequence at the ends. So lambda phases have single-stranded sequences. That is, although it is a double-stranded virus, that is double-stranded bacteriophage, that it has got both the strands of the DNA, which are complementary to each other. At their termini, they have staggered ends, they have unpaired ends, as we have seen in case of restriction endonuclease cutting, particularly that of type 2 restriction endonuclease cutting, that they cut fragments by the type 2 restriction endonuclease had sticky ends at their two termini. In this case also, though they have not been cut by any restriction endonuclease, it has been imparted by the nature and it has been gained as they have evolved. The bacteriophage, lambda bacteriophage, have got two ends having single stranded DNA stretching about 12 base pairs, and both of them are complementary to each other. That is, they are sticky to each other, they are cohesive to each other, and they cohere to each other, forming a circular lambda file. And that's why this site is called cos site or cohesive site. Then another feature which helps tremendously in employing the lambda file as a vector is that its central region, this region, from here to here, is devoid of any vital genetic information. That is, this region could be replaced by this stretch of DNA which we are desiring to clone. So here you can see 1/3 of DNA form continuous block in the middle of the genome, which could be manipulated without phage losing its ability to grow typically. So this stretch, which is in the middle, is devoid of any vital genetic information. That is, if we remove this stretch of DNA, the virus, the multiplication of phage will not be adversely affected. And that's why this provides such a platform for integration of the DNA of our choice. So it has got cos sites as well as it has got a platform, a location, a slot at which the desired DNA could be integrated. But another handicap is there. That is its peculiar characteristics in packaging, particularly in the morphogenesis of its heads. What happens that the morphogenesis of head is completed only when there is a minimum and maximum number of nucleotides in the virus, in the bacteriophage. So while replacing this region, we will have to see that whether the integrated DNA conforms to the maximum and minimum. Maximum is 105% of the normal DNA stretch of the phage. That is, the recombinant DNA should not exceed more than 105% of the length of the normal bacteriophage. So if the normal, say for example, I'm citing this fictionary example totally to make the thing clear to you, that say, for example, if the virus is 100 centimeter, viral genome, bacteriophage genome, lambda-phage genome is 100 centimeters long, the genome along with the integrated DNA, foreign DNA, should not exceed 105 centimeters. Similarly, it should not be less than the 60% of the genome, about 105% and about 105% and 60%. So it should not exceed the length by 105%, and it should not have lesser than 60% of the length of the normal bacteriophage DNA. So while integrating, while utilizing this stretch, we should see that it should not be longer than 105% of the length of the normal bacteriophage, lambda-phage, because then the assembly of head won't take place. Similarly, if it is less than the 60% of the normal stretch of the DNA of the lambda-phage, assembly of head won't take place again. So this is very important characteristic that we should keep in mind, that while integrating the DNA or while substituting a portion, we should see that it should not be less than the 60% of the length of the normal phage DNA, and it should not exceed the 105% of the length of the normal phage DNA. So its important characteristic was its important characteristics could be recounted, that this phage can have two types of infections: temperate infection as well as lytic infection. In case of lytic infection, they enter into their multiplying cycle, they form their mini copies called cell lysis and get released to infect newer hosts. Whereas in case of lysogeny, they enter into the host genome, and for that part of the life cycle, they are called prophage, and prophage multiply along with the host genome and do not cause cell lysis. Then they have the cos site at their end, cohesive site, that is the single-stranded stretch of about 12 nucleotides, which coalesce with each other, which stick to each other, giving it a rounded shape. The vector can take up to 20 kilobases of gene of interest. That is the foreign gene, which could be integrated into this bacteriophage, should be of about 20 kilobase stretch. Now, there is M13 vector. This is of a group of vectors which is called FF phage. FF phage, that is F filamentous phage. These phages are filamentous in nature, having protein cover and a short stretch of DNA, tightly packaged, and their DNA had also, you can say, discarded all the excess luggage overboard. That is, they have been very nicely packaged, optimizing the length of the DNA. That is, they have only that stretch of DNA which is necessary for their survival, but vector stuff only 507 nucleotide intergenic sequence is manipulative. So here, since they have optimized their length of the DNA, we don't have the manipulative, a longer manipulative site, and we have only 507 nucleotide intergenic sequences in case of this M13 phage, which is a phage of F filamentous category. So the next slide is concerned with the replacement vector and insertion vectors. So in case of replacement vectors, what happens that we replace a certain stretch of DNA. We replace a certain stretch of DNA say for this example, here we have a stuffer region. And this stuffer region had been cut by the restriction endonuclease. And in place of this stuffer region, we have inserted a fragment of DNA which has been cut by the same endonuclease. And so it has got sticky ends and it easily sticks to the cut termini of the bacteriophage genome. And that's how we produce the recombinant vector with insert. So for inserting the insert, we have removed the region of the bacterial genome, which is called a stuffer fragment. So the question is, how will we identify it? That what is the marker which will lead to its identification? How is it possible to identify the recombinant plasmid? So this is the stuffer region, okay? And the type of organism is high frequency lysogenic organism, that is 99.9%. 99.9% of the phage entering into this system will undergo lysogenic cycle. The phage, 99% of phage entering into this system will undergo lysogenic life cycle. And if lysogenic life cycle is pursued, in the lysogenic life cycle, we have already seen that the phage genome gets integrated into the host genome and it multiplied along with the host genome as if it were the part of the host genome. And so new phage will be produced. That is, the phage multiplication will not be pursued. Rather, phage will lie quiescent in the host genome. So when phage multiplication will not proceed, how will we obtain or harvest the recombinant DNA that we have inserted into the vector? So what happens that this gene for imparting it high frequency lysogenic characteristic lies in the stuffer region. Okay. So when we remove this stuffer region, what will happen? This gene will also be removed. Okay. So when this gene will be removed, this high frequency lysogenic property will also be removed. Is it not? So when we integrate it here, it has got integrated. What do you think? Whether it will pursue the lytic cycle or lysogenic cycle? since it was an HFL organism, that is high frequency lysogenic organism, it should have pursued lysogenic cycle, that is, it should not have made its own copies. But what happens in reality? Since that characteristic, that is the HFL characteristic, was imparted by a gene which was located in the stuffer region, and since this gene has been removed, it won't pursue lysogenic life cycle, rather it will pursue the lytic life cycle. So the lysogenic, high frequency lysogenic organism, when it starts producing phage population, that is, when it lyses and releases phage population, that is the indicator that the phage population is having this replacement, this inserted complementary gene rather than its own gene. So the removal of this segment imparts a selectable marker, that is, it acts as a selectable marker by which we select the transformed organism. So here we have seen the replacement vector, and now in this case there is insertion vector, that the vector is carrying a portion of foreign DNA which had been inserted at the desired locus inside it, and it has not been inserted by replacing the phage DNA, a segment of phage DNA. So here you don't have the stuffer region. So how will we identify this after the transformation, after the integration of the desired set of the DNA? So there is a P2 phage. This P2 phage is unrelated to the lambda phage. But when it is present, the lambda phage will not enter into the solution, will not enter and multiply in the solution. So the locus, and this sensitivity is imparted by a gene which is called PSI gene. So this PSI gene imparts the sensitivity to the P2 phase, and when this P2 phase is present, the lambda will not infect. The wild type lambda will not infect and will not pursue its life cycle. So this PSI is located here. This PSI is located here. Okay. So when you are cutting this fragment, cutting this side, the PSI is most likely to be disrupted. So the PSI will be disrupted and a new fragment of DNA has been integrated here. This integration causes disruption of the PSI. That means this lambda phage, which was not multiplying itself in presence of the P2 phage, will now multiply itself in presence of P2 phage. So the identification marker or the selection marker or the selectable marker in this case is the disruption of sensitivity imparting region of the gene, due to which the P2 phage inhibited the lambda phage. When the insertion is done in that sensitivity imparting region, the sensitivity is lost and the lambda phage will multiply and lyse the cell. So that is how we can identify a cell which is having the genetic new genetic marker inside it. So in this two types of vector, replacement vector as well as insertion vector, the selectable markers can be employed in different ways that we have seen. Now the next slide. It's cosmid. Cosmid is having cost slide. There was one important point here which I missed, is that while integrating this or this fragment, this stretch of DNA, we will have to keep in mind what? Our bar on the longest segment and the shortest segment. That is, the after integrating this replacement, this complementary DNA, the length of the phage should not exceed more than 105%, or after removal it should not decrease less than 60%. So the lower level is 60% and the higher level is 105%. So while integrating the new stretch of DNA, we should have an eye on this upper and lower limit of 105% or 60%, because if the size goes below 60%, the head will not be assembled, and if the size goes above 105%, the head will not be assembled again. So here, nothing special. We have got this we have seen that in the lambda phage, the both unpaired ends are called coside. There are about 12 unpaired or overhanging end in case of lambda phage, and they are called coside because they come to coalesce, their cohesive side, they come to get attached with each other. And this cos site is also important for the assembly of the lambda phage. So when we integrate this cos site into a plasmid, this plasmid will act as a cosmid. Plasmid having cos site, and this cos site will impart lambda-like characteristic to that plasmid. So that plasmid with integrated DNA can very much assemble as a lambda phage and multiply as a lambda phage and infect another bacteria as lambda phage were doing in natural case. So this is a synthesized plasmid having cos sites of the lambda, and so this is called cosmid. So hybrid between plasmid and lambda phage, lambda vector. So cosmid is nothing but it is hybrid between the lambda phage and the plasmid. Plasmid having cos side of lambda phage are called cosmids, and they can be packaged into infectious bacteriophages lambda. So these plasmids act as if these were the genomic constituent of lambda, because they get packaged into the lambda and lambda becomes infectious, infect another host and inject these cosmids into those hosts. So we can have the multiple copies of the DNA, stretch of DNA, which had been integrated into this cosmid. So we can carry, or the cosmid can act as a vehicle for 30 to 40 kilo base insert fragment. That is, an insert stretching about 30 to 40 kilobase could be integrated into the lambda file. An example is 2RB, which is the example of a lambda file. Cosmid. So here you see that how cosmid is in action. Here you have a cosmid. It has got origin site, and these are the restriction sites. Here we have got cos sites to cos site, and here the ampicillin resistance site. So it has been cut by a restriction endonuclease, and then here the restriction endonuclease is cutting, and then we have a new stretch of DNA. This stretch of DNA, it has been integrated here and sealed by T4 DNA ligase. We have studied it in detail in case of the previous section while we were dealing with the enzymes which were employed in case of recombinant DNA technology. And that's how this DNA is being packaged into the lambda file, as if it were the DNA of the lambda file. So here you have got plasmid with cos sites acting as cosmid with integrated DNA. And so this is the recombinant DNA which had been packaged into the lambda file due to the presence of cosmid. Had there not been cos site, they would not have been packaged. So we have imparted a plasmid, the packaging power into the lambda file by ligating cos sites to that plasmid. And when we ligate cos site, we integrate cos site to a plasmid, the plasmid becomes cosmid. Now, there is another vector which is called phosmid. So this is F factor plasmid. We have seen earlier in detail that fertility factor plasmid, which have got episomal characteristic too, because they get integrated into the host genome too. So this F factor plasmid gets integrated with the cos site, and that is called phosmid. Here you have got phosmid, here lactate, identifiable marker that we have discussed earlier, that how lactate carries out the synthesis of beta-galactosidase and how beta-galactosidase imparts coloring or property by converting BCIG into the indigo. And here we have cos site, here partition site, part A, part B, part C. So these sites are for segregation of the dividing phosmid vector. So the phosmid is F plasmid along with the cos site, which has been integrated here behind my picture. So the cos site integrated into a F plasmid gives the name phosmid. And larger stretch of DNA can be harbored in these phosmids, 100 kilobases. Now, Sharon vector. This Sharon vector is also a lambda bacteriophage, have been developed as vectors for cloning by engineering lambda bacteriophage. And in this Sharon vector, the common example is Sharon 40, 16 restriction enzyme possibilities. There are possibilities of creating sites for 16 restriction enzymes. And so we can integrate different fragments cut by different restriction enzymes, and that's how we imparts plasticity to carry different types of DNA fragments. You can see here, this side, this stuffer segment has been removed, and this side has been integrated, having multiple cutting side, and here you can integrate so many DNAs of your desired choice, and up to 24 kb kilobases stretch of DNA could be integrated. So replacement vectors with polylinkers. In the last slide, we have seen that we were replacing a chunk of DNA of the vector, that is the lambda bacteriophage, by the DNA which was to be propagated. Here, we remove a chunk of the host genome, of the vector genome, by polylinkers, and then we add various stretches of DNA into this polylinker, and we could then transform this Sharon vector into a multiple recombinant vector, that is a vector having multiple stretches of DNA from the different sources. So it will go a long way in cloning. Then there is phasmid. As the name indicates, it has got phage as well as plasmid characteristic. So it is a chimera. That is, it is having characteristic, it exhibits characteristics of two different organisms. In chimera, here, as a vector, it is exhibiting characteristics of two different vectors, that is, it has incorporated in itself the features of two different vectors. So it is a chimeric vector, and two different vectors are plasmid as well as the FF vector, F factor fibrous vector, which I have dealt with earlier, and that is F1, FD, or M3, having the origin sites of both. So the phasmid, it's chimeric in the sense that it carries the origin site of F1 vector, F1 phage. The examples are F3, FD, FN, F1. So it is carrying the origin site of phage as well as carrying the origin site of the plasmid too. So it is chimera of plasmid with single-stranded DNA phage such as F1, FD, and M3, having origin sites of both. So it has got single-stranded origin site as well as the double-stranded origin site. The double-stranded origin site was that of the plasmid, whereas the single-stranded origin site will that be of the phage which has been incorporated into it. And so it is a synthetic plasmid which is having the characteristic of both phage as well as plasmid, and it is called phasmid. So you see here that when this site will be activated, it will go phage way. That is, it will form replication here and form single-stranded DNA copies, because this is the origin site for the single-stranded DNA of F1, FD, or M3. And when this ORI site will activate it, this is the ORI site or origin site of the plasmid. So when this is activated, it will go plasmidy way. That is, it will act like a plasmid when this site will be activated, or it will act like a single-stranded bacteriophage origin site when this site will be activated. So it is a chimera between the single-stranded FF phage, which is F1 phage, FD phage, or M13 phage, and a normal plasmid. And since it incorporates characteristics of both single-stranded phage as well as the double-stranded plasmid, it has been named phasmid. Now you can see here the different location sites of the plasmid, coat protein site, plasmid site. But one more important point is that this plasmid contains gene for only one coat protein or none of the coat proteins. So how will be the assemblage of bacteriophage take place? When there is no gene for the cover protein, coat protein, then how would the virus assemble itself? Any guess? No. So we see here that to create maximum stretch, to create a bigger slot for the insertion of the DNA of the choice, all extra luggage had been removed. That is, the DNA stretch which was coating for the coat protein had been removed altogether, or only a few proteins coding genes have been left there. Then if the coating proteins will not be coded, how will the virus assemble? How will the phage assemble? And for that purpose, we introduce a helper phage. And the helper phage has been manipulated in such a way that its oocyte will not function properly, and that's why it will not form its own copy. Rather, it will only synthesize the proteins which were coded for the coat proteins. And so the coat in this plasmid, the stretch of DNA, a longer stretch of DNA will be integrated in the plasmid, but the assembly will take place only in presence of a helper phage, which will not multiply itself, but which will code for the coat proteins, and the coat proteins coded by the helper phage will be incorporated at the coat protein of the plasmid, and that's how the new plasmid containing organism will be given. That is the phage containing the new plasmid will come into effect. So here you have got this. This is the plasmid having the antibiotic resistance, plasmid origin, and filamentous bacteriophage origin. You see here two types of origin: plasmid origin and bacteriophage origin. And here is the DNA, a few peptide promoter region, and here you have the region in which the foreign DNA will be incorporated. And so this plasmid can multiply only when it takes shape as a bacteriophage, since it does not have the stretch of DNA which codes for all the proteins which are incorporated while the assembly of the bacteriophage is taking place. It will not itself morphogenate into a bacteriophage, and there it will have to take the help of super infection with a helper phage. That helper phage has been deliberated in such a way that it is unable to multiply itself, but it will regulate the synthesis of coat proteins, and those coat proteins will be implied by this plasmid for the assembly as a phage, and so this assembly as a phage has taken place. That's how we have come to the today's lecture, come to the end of today's lecture, which discussed in quite detail the different types of vectors used for propagating the desired DNA into different platforms for their cloning or for their expression. We have seen that how vectors can be expression vectors, they can be cloning vectors, they can be chimeric vectors, they can be self-dependent vectors such as plasmids, and how different plasmids have been synthesized by incorporating polylinkers, that is multiple integration sites on which DNA of desired choice could be integrated. That is, the slot creation had been done in artificial way so as to integrate longer stretches of DNA and various stretches of DNA. That is, they do not vary only in length, but they also vary in type too. So these vectors are very important in giving effect to our technology, that is recombinant DNA technology. The vector story has not come to end as yet, because we have devised artificial chromosomes too, and we have got yeast artificial chromosome, we have got bacterial artificial chromosomes, we have got human artificial chromosomes too. So we have BACs, HACs, and YACs that you can say as acronyms. So we shall be discussing